Welcome to Lecture 3.1, in which we will begin discussing cells and cell theory. This over here is a video from early in my PhD, featuring beta cells which release insulin in response to glucose to maintain our blood glucose levels. You can see the nuclei of each cell, and how these cells are all flashing green together, which represents a response to glucose. If you're curious and would like to learn more, feel free to come talk to me. Our learning objectives for this video are to defend why a cell is the basic unit of life, list the three components of cell theory, understand why the surface area to volume ratio is important for cells, and describe the four features found in all cells. Cells are the smallest unit capable of presenting all of the characteristics of life. A simple bacteria may only have one cell, but it can still perform metabolism, sense and respond to its environment, and maintain its internal environment through homeostasis. On the other hand, macromolecules cannot be living because they are by themselves unable to do these things. DNA, for example, makes up our genetic material, but cannot perform metabolism or maintain homeostasis. Organelles that make up eukaryotic cells are unable to live outside of the context of the cell, because they would not have all the components they need to survive. There are many types of cells, and even within the human body, we have many cells that have distinct features that allow each cell type to carry out its designated function. Since they're so small, most cells require a microscope to be seen. The first recorded observation of cells was in 1665 by Robert Hooke, who was looking at corks under a microscope he built. This image was drawn by him in his book Micrographia, which was the first major publication that contained a scientist's observations made using a microscope. Each of these boxes is an individual cell, and he actually coined the term cell because their box-like appearance reminded him of small rooms in the monastery referred to as cells. Several other scientists also made huge discoveries that contributed to our understanding of the cell, and almost 200 years later, the cell theory was proposed. This theory has three main ideas. 1. Cells are the basic unit of life, as we previously discussed. 2. All living organisms are composed of one or more cells. That is, organisms can be unicellular or multicellular. 3. All cells come from the reproduction of existing cells. While the first cell is thought to have formed spontaneously, all cells that have been observed have been derived from other existing cells. We will discuss cell reproduction further in a later lecture. Why are most cells small? It has to do with the ability of the cell to exchange nutrients, oxygen, and waste with its environment. As size increases, the distance that materials need to travel increases as well. Let's look at this 1-inch cube representing a cell here. In the center, we have the nucleus. The distance between the membrane of the cell and the center of the cell is pretty small. Now here is a larger 2-inch cell. The distance between the membrane of the cell and the center of the cell is now twice as large, so it would take longer for materials to enter and leave. Since nutrients, oxygen, and waste are exchanged with the plasma membrane, the surface area of the plasma membrane is also important, because more surface area allows for better absorption and excretion. However, as size increases, surface area decreases relative to the volume. Going back to the 1-inch cube as an example, the surface area, abbreviated as SA, is 6 square inches. The volume, abbreviated as V, is 1 cubic inch. The surface area to volume ratio is 6 divided by 1, which gives us 6. Now let's look at the 2-inch cube. The surface area is 24 square inches, which is larger overall, but since the volume is now 8 cubic inches, the surface area to volume ratio is now only 3. A lower surface area to volume ratio means that exchange of materials across the membrane is less efficient. Now let's look at the 2-inch cube that's made of multiple 1-inch cubes. There are 8 1-inch cubes here, for a total surface area of 48 square inches, and a total volume of 8 cubic inches. Now it has the 2-inch cube's volume, but the surface area to volume ratio of the 1-inch cube. This allows for more efficient exchange while maintaining a larger size, and is why large organisms tend to be multicellular. Some cells have developed different strategies to overcome the surface area to volume ratio problem. For example, intestinal cells have many little folds in the membrane that increase the surface area for better absorption of nutrients. Neurons have a more elongated shape, which also helps increase surface area. Muscle cells have multiple nuclei so that they can get better access to oxygen and nutrients. Now let's talk about the light microscope. Here is an image of a light microscope with various parts labeled. At the top, we have the ocular lenses, which are also referred to as eyepieces. This is the part we would use to look into the microscope and observe our sample. The ocular lenses generally have a 10x magnification. The objectives are over here, and we can rotate between the objectives using the nosepiece. Many light microscopes have 4 objectives, with 4x magnification, 10x magnification, 40x magnification, and 100x magnification. The total magnification is equal to the magnification from the ocular lenses multiplied by the magnification of the objective being used. For example, the 4x objective would have a total magnification of 40x. You will get to work with the microscope and learn more in the lab. Light microscopes have a limited resolution of 200 nanometers, which means that it cannot distinguish 2 objects that are within 200 nanometers of each other. That's because these microscopes are limited by the wavelength of white light. Electron microscopy was invented in the 1930s and allows for much higher magnification. Instead of light, it uses beams of electrons. There are 2 types. Scanning electron microscopy allows for a resolution of 10 nanometers, and is often used for scanning the 3D surface of organisms in high detail. Transmission electron microscopy has an even higher resolution of 0.2 nanometers, and can be used for observing very fine details within cells. This slide is just a reference for the scale of life. The scale is logarithmic, which means that each tick mark represents a 10 times difference in size. We can see quite a few things just with our eyes, but would need a light microscope to see anything smaller than 100 micrometers, such as cells and some organelles. An electron microscope is needed to see things that are even smaller, such as macromolecules. Here are some examples of microscope images. On the left, we have what is called an H&E stain of cancerous kidney cells. The H stands for hematoxylin and stains the cells pink and the surrounding connective tissue a lighter pink. The E stands for eosin, which stains the nuclei purple. In the center, we have a fluorescent image of root cells found in a flowering plant. The nuclei are stained by DAPI, which is a dye that strongly binds to DNA and stains the nucleus blue. The green is a component that is found in the cytoskeleton of all cells, which we will talk more about later. You can see that it is all over the cell, but also has some stronger intensity around the membrane of the cell. This is a scanning electron microscopy image of E. coli, and you can see that the scale bar is for a much smaller amount here. In fact, this image was taken at over 6,000 times magnification. There are a few things that all cells have in common, whether they are prokaryotic or eukaryotic. Every cell must have a plasma membrane that separates the cell from its external environment, a cytoplasm that makes up its internal environment, genetic material in the form of DNA, and ribosomes that serve as protein-making machines. This diagram also shows RNA, which is made based on DNA. In prokaryotes, DNA is found in the nucleoid region, while in eukaryotes, the DNA is compartmentalized into a separate structure called the nucleus. We will talk more about the differences between prokaryotic and eukaryotic cells over the next 2 videos. In this video, we learned about the cell theory, which states that cells are the basic unit of life, all living organisms have 1 or more cells, and every cell comes from a pre-existing cell. Because cells are so small, it wasn't until light microscopy was invented that their existence was discovered. The reason why they are so small is because it helps them maintain a good surface area to volume ratio that allows for efficient exchange of materials in and out of the cell. Although cells can be prokaryotic or eukaryotic, all cells have plasma membrane, cytoplasm, genetic material, and ribosomes. In the next video, we will begin talking about prokaryotic cells. Thanks for watching.